Hello and welcome to the channel to help you ace your GCSE Combined Science exams. Note, this is information primarily from the OCR Gateway exam board, so if you are taking another one, make sure to check this information is relevant to you. And with that, let's get on with today's video. In today's video, we will be going over B1 Cells and Microscopy. Biology is all about living stuff, and all living stuff contains cells, so let's start there. You will need to learn the structures within eukaryotic cells and prokaryotic cells. Eukaryotes are animal and plant cells containing similar subcellular structures. This is the nucleus. It contains DNA in the form of chromosomes that control the cell's activities. These are mitochondria. They are the site of cellular respiration and contain the enzymes needed for the reactions involved. This is the cytoplasm, a gel-like substance where most of the chemical reactions will happen. These are ribosomes. Ribosomes are a cell structure that make protein. Protein is needed for many cell functions such as repairing damage or directing chemical processes. The final structure is the cell membrane. This holds the cell together and controls what goes in and out by providing a selective barrier. They also contain receptor molecules that are used for cell communications. Moving on to plant cells, these have everything animal cells have, plus a few others. They still have the nucleus, cytoplasm, mitochondria, ribosomes, and the cell membrane. However, there are now 3 new additions to this cell that you will need to know the structures for. First, the cell wall. This is made of cellulose, which gives support for the cell. These are chloroplasts. They are where photosynthesis occurs and contain a green substance called chlorophyll. And finally, this is the vacuole. The vacuoles tend to be very large and are extremely important in providing structural support as well as serving functions such as storage, waste disposal, protection and growth of the plant. Next up are prokaryotes. These are bacteria and are much smaller and simpler cells. Just like in eukaryotic cells, prokaryotic cells also contain a cell membrane which controls what goes in and out and is also supported by a cell wall. The chromosomal DNA is one long circular chromosome that controls the cell's activities and replication. It floats free in the cytoplasm and is not in a nucleus. Plasmids are small loops of extra DNA that aren't part of the chromosome. Plasmids contain genes for things like drug resistance and can be passed between bacteria. Now we know about cells, it's time to find out how we study them. Cells are studied using microscopes. For the OCR Gateway exam board, you will need to know 2 types of microscope. These are electron and light microscopes. Microscopes use lenses to magnify images. They also increase the resolution of an image. This means they increase the detail you can see. Resolution is how well a microscope distinguishes between 2 points that are close together. Light microscopes were invented in the 1590s. They let us see things like nuclei and chloroplasts. We'll learn more about the structure of these in the next video. Moving on to electron microscopes, these were invented in the 1930s. They let us see much smaller things in more detail, like the internal structure of mitochondria. This has allowed us to have a much greater understanding of subcellular structures. Only electron microscopes will let us see things as tiny as plasmids or viruses. Transmission electron microscopes, or TEMs, have a higher magnification and resolution than light microscopes, but they're not portable. They're expensive and it's a complicated process to prepare specimens for use, which means they can't be used to look at living tissue unlike light microscopes can. And there we go. Now you know all the structures within inside prokaryotic and eukaryotic cells, and you know the difference between light and electron microscopes. If you did enjoy the content covered in this video, make sure to drop a like and hit that subscribe button, and I'll see you in the next video.